Well, hello, everyone. I'm George Koob. I'm the director of the National Institute on Alcohol Abuse and Alcoholism of the National Institutes of Health. And I also have a laboratory as a senior investigator in the Integrative Neuroscience Research Branch of the Intramural Research Program at the National Institute on Drug Abuse. I'm going to talk today about the neurocircuitry of alcohol use disorder. The outline of my talk is I'm going to talk about a heuristic framework for research on the neurobiology of alcohol use disorder. That'll include a scope of the problem, the three-stage cycle of addiction, positive reinforcement, opponent process, hyperketifia, and negative reinforcement. And I'm going to drill down into the binge intoxication stage and talk about the neurocircuitry mediating domains of alcohol reward, incentive salience, and pathological habits, followed by drilling down into the withdrawal negative affect stage, the neurocircuitry mediating domains of reward deficits and stress surfeit. And finally, the preoccupation anticipation stage, the neurocircuitry mediating domains of craving and executive function deficits. But before we get too far, I'd like to talk just a little bit about the scope of the problem. I'm going to be focusing on alcohol today, but I want you to realize, of course, we have an opioid crisis. But when you look and compare alcohol to opioids, you see that while there are 2 million individuals with opioid use disorder, and that has been increasing, we always have an endemic problem of about 14 million individuals with an alcohol use disorder. And alcohol use disorder endemically causes now 95,000 deaths annually, everything from car accidents to contributing to overdoses. And I might add that alcohol is responsible for half of liver disease in the United States, half. And the alcohol also plays a prominent role in what we call the deaths of despair. And the right-hand panel shows you some of the classic work of Ann Case and Angus Deaton, showing that over the last basically 15 years, there's been increases in mortality. And their initial study was on white non-Hispanics, age 45 to 54. These were increases in poisoning and suicides and chronic liver disease, all of which alcohol contributes to these diseases. And subsequently, these patterns of mortality have now been observed across many racial and ethnic and age groups. And alcohol-related deaths actually doubled between 1999 and 2017. So these statistics, along with other recent reports that have highlighted changing trends in drinking patterns, increasing consequences of alcohol in two populations, women and the aging population. So what is addiction? What do we mean by addiction? Or what do we really mean by a moderate to severe alcohol use disorder? And we know of alcohol use disorder as a spectrum disorder at this point. And according to the American Psychiatric Association Diagnostic and Statistical Manual, alcohol use disorder can range from mild to moderate to severe. But when we talk about addiction, we're usually talking about the more moderate to severe alcohol use disorder. And we can define this as a chronically relapsing disorder that is characterized by a compulsion to seek and take drug or stimulus, loss of control and limiting intake. And everyone agrees with that definition. But I have an add-on, which is the emergence of a negative emotional state, dysphoria, anxiety, irritability when access to the drug or stimulus is prevented. And I've defined this over the years as the dark side of addiction. Today, I'm going to talk about all three stages of the addiction cycle as we have heuristically defined them. I'm going to talk about the binge intoxication stage and incentive salience and pathological habits. And I'll talk about the withdrawal and negative affect stage, which includes the domains of reward deficits and stress surfeit. And I'm going to talk about the preoccupation anticipation stage, or what we commonly call the craving stage, which encompasses executive function deficits. And these are all color-coded on this slide by different circuits: the basal ganglia for the binge intoxication stage, the withdrawal negative affect is the extended amygdala in red, and of course the craving or preoccupation anticipation stage is in green here, which is illustrated by largely prefrontal cortex, but also some old cortex such as hippocampus and insula. But when I talk about that add-on to the definition of addiction, it brings me to what are the neuroadaptations that occur in these circuits? How do these circuits change as addiction progresses, as an alcohol use disorder becomes more severe? And this goes back to an old theoretical framework from Dr. Richard Solomon at the University of Pennsylvania. In simple terms, it's called opponent process, or what goes up must come down. The title of this paper was actually a standard pattern of effective dynamics produced by novel and repeated unconditional stimulus. So if you look at this slide, you can see that there's this diagram here of an A process followed by a B process. And this is supposed to be hedonic state. So an A process is actually feeling good. And you could argue that the B process is feeling bad. And in this particular theoretical illustration, Solomon was talking about actually heroin administration intravenously. And you can see there's a hedonic response and then a dysphoric B process response. You could think of this in terms of a binge on alcohol as well, whereas the A process would be the binge and intoxication, the B process would be the hangover the next day. And that happens in a non-dependent individual, a person without alcohol use disorder. But if you keep drinking and you drink and you drink and you drink, you end up dependent on alcohol in the old sense of the term, which means that there's a withdrawal syndrome manifested when you stop drinking. And that means also the development of tolerance to the hedonic effects. So you lose the A process, but you gain the B process. And we're going to argue that the B process actually subtracts out the A process. And that B process is going to be what I talk about as the negative emotional state that's associated with removal of alcohol when you're dependent. And we have actually made up a word to describe that state, which is called hyperketifia. And it's defined as the increased intensity of negative emotional, motivational symptoms and signs observed during withdrawal from abused drugs. It's derived from the Greek word ketifia for dejection, sadness, a negative emotional state. It's hypothesized to represent elements such as dysphoria, irritability, alexithymia, or simply symptoms often described as ill at ease, uncomfortable within one's own skin, or simply not hedonically normal, symptoms historically difficult to define. What I was really looking for was I was tired of saying the word hypersensitive negative emotional state associated with withdrawal from alcohol and opiates. And so I called my brother, who was fluent in Greek and a conservator in archaeology, and I asked him for a word that defines sadness in Greek. And I was thinking of hyperalgesia, which is increased sensitivity to pain. And I wanted a word for increased sensitivity to emotional pain. And thus we came up with hyperketifia. And this begs another theoretical framework that we need to discuss. And that is, we'll talk a lot about positive reinforcement. We always do. And that's defined as the process by which presentation of a stimulus, the drug here being alcohol, increases the probability of you drinking again. But there's another source of reinforcement that hyperketifia contributes to, and that is negative reinforcement. And that's defined as the process by which removal of an aversive stimulus, the negative emotional state or hyperketifia of drug withdrawal, increases the probability of response. And this is variously described as dependence-induced drug taking, or in some sense, self-medicating the condition that alcohol itself caused. So you may well ask, well, where did these stages come from and where do these domains come from? And are they really of relevance to the true phenotype of alcohol use disorder? And so at NIH a number of years ago, Laura Quacco led our team there in describing a framework for improved diagnosis, prevention, and treatment of alcohol use disorders. We call this addictions neuroclinical assessment. And she hypothesized, we all hypothesized, that the three stages could be represented by these three domains: incentive salience, negative emotionality, and executive function. And perhaps one could develop and measure tests that are scales of how you feel about things, but also tests that are actually operational tests, such as the Q reactivity test for incentive salience, or the facial emotional matching test for negative emotionality, or delayed discounting. And so the goals of the ANA were to identify how these three domains influence differences between people diagnosed with AUD, which may then be used to guide treatment decisions and to better understand differences between individuals with and without alcohol use disorder. But one question is can we actually validate these three stages using this kind of approach? And so in a study that was published in 2019, showed that the three neurobiological domains hypothesized to be critical to the addiction cycle, incentive salience, negative emotionality, and executive function, could be identified through factor analysis in a large, diverse clinical sample representing the spectrum of alcohol use disorder. So these measures were taken of addiction, personality, cognition, behavior, and exposure, and early life stress. And they were collected in 454 patients from 2015 to 2016 at the clinical center at NIH. The study confirmed the relevance of these three neurofunctional domains to alcohol use disorder. And using multiple indicators, multiple causes, which is called the MIMIC approach, early life stress and socio-democratic predictors were identified. And you can see this in the diagram on the right. So we were able to validate these three domains in a human population of alcohol use disorder. And I might add that this study has now been replicated several times with ongoing studies. So this all boils down to a framework of the etiology of addiction on alcohol. And basically, we're not denying that reward brings you to the cycle. And that's the positive reinforcement illustrated in the blue here. But there's also another source of reinforcement, as I mentioned, which has been variously described as relief, or I call it negative reinforcement. And then eventually you may stop. You may run out of alcohol. You may reach the point where you're in severe withdrawal and you decide to do something about it, but you enter a stage known as protracted abstinence, which can feed back and lead to relapse. And so I would argue that this loss of control and compulsivity that we associate with alcohol use disorder is derived from three sources, not one source, but three sources. The development of incentive salience and pathological habits for drugs, the development of reward deficits and sensitization of stress neural circuits, and compromised executive function. So what are these circuits and how do they change as we move through the addiction cycle? So let's start with the binge intoxication stage, which has been a very popular area for research in the neurobiology of alcohol addiction and alcohol use disorder. There's been literally hundreds of studies on why alcohol is reinforcing. We know that it involves structures in the basal ganglia, which are illustrated on this slide. NAC stands for the nucleus accumbens, DS stands for dorsal striatum. And we know two key neurochemical systems are dopamine and opioid peptides. And I love this diagram that Eric Nestler developed a number of years ago, and we've modified it over the years, but basically the core diagram remains that the basal ganglia, particularly the nucleus accumbens, involves a dopamine input to the nucleus accumbens, but also an opioid peptide input to the basal ganglia. There is opioid peptides can interact directly with the dopamine system or independently and activate the nucleus accumbens. And so this ventral tegmental area, nucleus accumbens pathway, but also an independent opioid peptide pathway, is the circuit that is thought to mediate the rewarding or pleasurable effects of alcohol. And there's pretty good evidence, even in humans, that dopamine plays a role and is activated when you become intoxicated. So in individuals that received a dose of 0.75 grams per kilogram, about 3 standard drinks of alcohol and a cranberry juice and soda preparation. In this study from Yale University, published in Biological Psychiatry, you can see that when intoxicated, there's a decrease in dopamine D2 receptor binding that suggests that endogenous dopamine is being released that displaces the binding to this receptor. And it's more dramatic in males than it is in females and correlates better in males than it does in females, as you can see in the line diagrams here in this figure. So what about animal studies where we've tried to really understand specific neurocircuitry? Well, there are a whole variety of animal models that are thought to be relevant to the reinforcing effects of alcohol. And these include intravenous and oral self-administration and binge drinking paradigms, and brain stimulation reward facilitation, and conditioned place preference, where an animal returns to the place where it received alcohol. In the old days, people used drug discrimination to isolate compounds that may be like alcohol and not like alcohol. There's genetic models of high drinking, and there's drug taking in the presence of aversive consequences, which is a very popular technique now for measuring compulsivity in alcohol use disorder. And you can see here that animals will drink huge amounts of alcohol, particularly C57 mice in a particular paradigm called drinking in the dark, where they are allowed a limited access of 2 hours access on days 1 to 3, and then 2 or 4 hours access on day 4, the beginning of the dark period when mice are active and normally eating. And you can see that naltrexone dose dependently reverses this binge intake and resulting blood alcohol levels. And I just want to remind you that, again, we can see the same things in human studies. This classic study by Jennifer Mitchell, published in Science Translational Medicine, when someone was drinking the equivalent of one standardized drink, when individuals in a PET scan using carfentanil, a very, very potent opiate, a label, you can see, again, similar to what we saw with the release of dopamine, there's a release of endogenous opioids in the nucleus accumbens, both on the left side and on the right side. Again, the release of these endogenous opioid peptides is displacing the binding to the mu opiate receptor. And just to kind of pull a wrap on this, in a sense of translational value, we know from a whole series of studies that in primates, naltrexone will decrease alcohol self-administration. And in the two bottom panels, you can see that naltrexone decreases relapse to drinking and helps individuals maintain abstinence from drinking treated in human studies. And these were two early studies by Jovo Pacelli and also Stephanie O'Malley that led to the approval of naltrexone as a treatment for alcohol use disorder. And as most of you know, naltrexone is available either orally or by a long-acting preparation known as Vivitrol. So this is the rewarding effects of alcohol, and has led to one of the prominent pharmacological treatments for alcohol use disorder. But there's an evolving discussion of then what happens? What happens in this circuitry once the rewarding effects take place and the nucleus accumbens is activated? And this brings us to incentive salience. And incentive salience can be defined as the motivation for rewards derived both from one's physiological state and previously learned associations about reward that is mediated by the mesocortical limbic dopamine system. I would add that probably the opioid peptide system also contributes, certainly for alcohol, to incentive salience. The construct of conditioned reinforcement preceded and laid the foundation for incentive salience. So a cue that's paired with a reinforcer can come to elicit a response, a pleasurable response, or an activating response similar to the primary reinforcer. But incentive salience and conditioned reinforcement can be defined as a strengthening of behavior by a previously neutral stimulus through its association with the primary reinforcer. And that such becomes a reinforcer in its own right. So animals will actually work for the cue that was paired with something pleasurable, and so will humans. But both conditioned reinforcement and incentive salience provide constructs for what underlies cue-induced drug-seeking self-administration behaviors and conceivably the transition to habit-like compulsive seeking. So the incentive salience part is that the cue not only can produce the same effects as the primary reinforcer, but can motivate one to seek the primary reinforcer. And so to put it in very simplistic terms, when a honeybee sees a flower from which it derives a good deal of pollen, it will circle back to the hive and tell all its friends, and then the incentive salience that's generated is they'll return to that flower to seek more of the pollen because they have remembered that cue, so to speak. And the same thing happens with drugs and with alcohol. But then what happens? Well, another theory that's prominent in the binge intoxication stage is that there is a shift from the nucleus accumbens that I've been talking about to the dorsal part of the basal ganglia. And this shift is driving elements of compulsive seeking behavior that derive from habits, the formation of habits. And so this comes from some of the early work of Suzanne Haber showing that there are cortical, striatal, palatal, thalamic loops, but also that the activation evolves from the ventral part of the basal ganglia, which is the nucleus accumbens, to the dorsal part of the basal ganglia. And Barry Everett and Trevor Robbins have argued that this helps to consolidate habits and make drug seeking more automatic and less thought through. And so individuals will be drinking, even though they may not even cognitively be aware that they're engaged in over drinking. So pathological habits I've defined as maladaptive incentive habits are hypothesized to result from pathological coupling of drug-induced influenced motivational states and a rigid stimulus response habit system. And as a result, drug-associated stimuli through automatic processes elicit and maintain drug seeking. Here, goal-directed actions are operationally defined as sensitive to reinforcer devaluation, and habits are resistant to reinforcer devaluation. Drug-seeking and taking habits in those individuals with addiction to drugs are also reinforced by Pavlovian drug-associated condition stimuli in the environment, where they act as condition reinforcers and support protracted sequences of behavior, often in the absence of outcome. And so in the alcohol field, neuroadaptations in the dorsolateral striatum have been hypothesized cortical striatal palatal thalamic loops in compulsive drug seeking. And so in this expose by David Lovinger and Tom Cash, the changes in the dorsolateral striatum in acute alcohol exposure involve a decreased initiation in GABA synapses, but increased outputs through the ventral pallidum. And in chronic ethanol, there's a long-term inhibition of GABA synapses and a loss of modulation of the glutamate synapses, resulting in increased glutamate release. And so there's a shift from GABAergic control of glutamate to a loss of GABAergic control. And these changes, increase glutamate release and decrease GABA inhibition, are argued to, quote, foster greater dorsolateral striatal output in response to a given set of inputs from sensory motor cortex, potentially facilitating habit formation. And there's a lot of data in animal studies that support this hypothesis in the alcohol field. So now let's move to what I call the dark side. Let's move to another source of reinforcement and another yet series of changes in neuroadaptation that involve the withdrawal negative affect stage. And this involves now the extended amygdala, which I'm going to argue is the central nucleus of the amygdala, the bed nucleus of the stria terminalis. And now I've turned the nucleus accumbens red, because the nucleus accumbens, the shell of the nucleus accumbens in particular, is an accessory area to the extended amygdala, and it shares neuroanatomical connections and neurochemical makeup with the bed nucleus, stria terminalis, and the central nucleus of the amygdala. And I'm going to argue that the negative affect, the hyperketifia, that's associated with the withdrawal negative affect stage involves not only a loss of reward function, as we'll talk about in a second, but also a gain in the brain stress system. Now, what about animals that are dependent? How do we do that? Well, there have been a whole variety of different methods used over the years to induce dependence in animals. You can use allow animals to drink alcohol for very long periods of time. You can use animals that are predisposed to drinking excessive amounts of alcohol. But one method that was developed at the Salk Institute many years ago by Joe Rogers and Sandy Weiner and Floyd Bloom was to induce dependence by exposing animals to ethanol vapor. And this has now been extended to both rats and mice. There's a picture here of an animal who was allowed to self-administer alcohol for 30 minutes a day. During withdrawal from chronic exposure to ethanol vapor, exposure to ethanol vapor that generates blood alcohol levels around 0.2 gram %. That's almost 2.5 times the legal limit. And you can see over here on the right that the animals will escalate their intake, and you have animals now drinking the equivalent of 4 to 6 drinks in a human measure in 30 minutes, the rats. And you can see the same on the bottom for mice who are drinking in a 2-bottle choice with also chronic exposure to alcohol. So using this approach, a whole series of studies have been done, but we have to take a little step back to the conceptualization of how things could change with chronic administration of drugs, but chronic administration of alcohol in particular. So a long time ago, Floyd Bloom and I hypothesized in science that there were 2 types of neuroadaptations that could occur. We defined them as a within system neuroadaptation and between system neuroadaptation. And they're illustrated on this slide, but let me just simply say a within system neuroadaptation would be a loss of function in the same circuitry that mediates feeling good, the reward effects of alcohol. Whereas a between system neuroadaptation would be recruitment of some other system to act in opposition to the feeling good. In other words, a feeling bad system. So what's the evidence? Well, a lot of animal studies over the years have shown that there are decreases in mesolimbic dopamine activity during ethanol withdrawal, usually measures in the nucleus accumbens with in vivo microdialysis, as you can see in the top panel from the work of Marco Diana. Ethanol withdrawal decreases dialysate dopamine concentrations in the nucleus accumbens, but also decreases VTA dopamine firing during withdrawal. And at the bottom, you can see animals that were maintained on an alcohol liquid diet, put into withdrawal, and they actually, when given access, will self-administer enough alcohol to return their dopamine system to normal levels. And you can see here, there's a decrease in dopamine during withdrawal, and they self-administer enough alcohol to reverse that decrease in withdrawal. And many of you may be already familiar with the seminal work of Nora Volkoff, who has shown that in individuals with alcohol use disorder, there's a decreased methylphenidate-induced dopamine release in the striatum. And so if you look at the control subjects up at the top here, you can see that, again, when they're given methylphenidate, there's a dramatic release of dopamine in the striatum because you see less binding. Again, you're displacing the endogenous ligand. But when you look at the bottom panel, you see that that does not occur in the subjects with alcohol use disorder. They show a blunted dopamine release to a challenge. What about the between system neuroadaptations? Well, much work has gone into this in the alcohol field in a whole series of domains at this point. One of the prominent was the identification in 1981 by Wiley Vale and the group at the Salk Institute that CRF, corticotropin releasing factor, is a neuropeptide in the brain that drives not only the pituitary adrenal response to stress, not only the sympathetic nervous system response to stress, but also behavioral responses to stressors. And subsequently, there's been a series of studies showing that alcohol withdrawal increases CRF, not decreasing, decreases dopamine, but increases CRF in the central nucleus of the amygdala. And CRF antagonists in animal studies can reverse withdrawal-induced anxiety-like responses, can reverse dependence-induced self-administration of alcohol, as I showed you earlier, and can reverse stress-induced reinstatement in animal models where stressors are used to reinstate drug-seeking behavior. And in a whole series of elegant studies, Dr. Olivier George, in collaboration with Dr. Giordano Di Guglielmo and Dr. Robert Messing, have been able to use a very special rat that expresses CRF-CRE in the central nucleus of the amygdala and the bed nucleus of stria terminalis. And they've been able to use halorodapsin to suppress the release of CRF in these CRF-CRE animals. And you can see that when they turn on the light, and they actually shine the light on the central nucleus of the amygdala, you see when the laser is on, it decreases the drinking of the animals. When the laser is off, they go back to regular drinking. And that can be replicated with shining the light and activating halorodapsin in the bed nucleus of stria terminalis, but not when the light is shown in the lateral hypothalamus or the substantia nominata or the parabrachial nucleus. And this leads to the hypothesis that there is probably a projection and interaction between the central nucleus of the amygdala and the bed nucleus of stria terminalis in driving the circuitry involving CRF activation during alcohol withdrawal. And then in another recent study, Dr. Patty and the Winder and Cash group have shown that alcohol dependence dysregulates a GABAergic microcircuit in the bed nucleus of stria terminalis in alcohol dependence. And you can see here the inputs to the bed nucleus of stria terminalis, but one of the prominent ones is, of course, the central nucleus of the amygdala, illustrated in the bottom picture here. But there's also outputs of the bed nucleus of stria terminalis to brainstem, and there are inputs from cortical areas. And so following 72 hours of withdrawal from 4 weekly cycles of chronic intermittent ethanol vapor in mice, the excitability of either VTA or lateral hypothalamic neurons was increased. And withdrawal from chronic intermittent ethanol also increased the excitability of non-CRF bed nucleus of the stria terminalis neurons that project to both the LH and the VTA. So withdrawal from chronic ethanol is hypothesized to dysregulate CRF GABAergic microcircuits that inhibit anxiolytic outputs of the bed nucleus of the stria terminalis, which may contribute to the enhanced anxiety during alcohol withdrawal and drive alcohol-seeking behavior. And you can see the changes in excitability of these neurons in the left-hand side of the panel here at the bottom. And so the argument would be that we've identified a very specific circuit involving CRF, but other neurotransmitters that project from the central nucleus amygdala to the bed nucleus stria terminalis, and then possibly on to the lateral hypothalamus and back to the ventral tegmental area to drive this motivated negative reinforcement form of compulsive drinking. And just to add another link to this that has translational potential, one of my former postdoctoral fellows, who's now a senior staff scientist at the National Institute on Drug Abuse, Dr. Leandro Vendescola, had noted a long time ago, and we had pulled from the literature, that glucocorticoids are activated during alcohol withdrawal. That's the classic hormonal stress response. And we know that those glucocorticoids are driven by CRF from the hypothalamus. And we know that then the glucocorticoids feed back and suppress this hormonal release by feeding back to the paraventricular nucleus. And that's shown on the red diagrams down at the bottom. And so I always say, why don't you become cushingoid when you're writing a major center grant or a training grant for NIH? And the reason you don't become cushingoid is because you have a protective mechanism, a homeostatic protective mechanism that feeds back and shuts off this massive release of glucocorticoids. But the part they leave out of the textbooks is, in fact, the glucocorticoids feed back and drive the amygdala, and they drive CRF in the amygdala. And so Leandro asked whether he could do this study where he gave chronic mefopristone, a glucocorticoid antagonist, and would that block this escalation in intake that we see in dependence? And you can see the data here, and you can show there's a dramatic blockade in the animals treated with mefopristone in their escalation in alcohol intake. And then Dr. Barbara Mason went on and showed that she could actually treat human patients in a human laboratory study with mefopristone, and not only block the craving that was associated with exposure to cues that signified alcohol, but that alcohol was unavailable in this human laboratory study, but also showed that 1 week after stopping the drug and 2 weeks in follow-up, these individuals drank less alcohol. And so this was published in the Journal of Clinical Investigation a few years ago. Again, illustrating the translational potential of some of these neurocircuit changes that we've been observing in the field over the last 10 to 20 years. And then I just want to mention that there's another neurotransmitter, another stress neurotransmitter in the extended amygdala that has a focal point in not only the central nucleus of the amygdala, but also the nucleus accumbens, and there's also dinorphin in the frontal cortex. And a long time ago, Bill Carlizon and Eric Nestler again hypothesized that if you excessively activated dopamine receptors or opioid receptors in the nucleus accumbens, you drove a biochemical process by which cyclic AMP was activated, cyclic AMP response element protein was activated, and dinorphin was activated, and this dinorphin fed back and shut off the dopamine system. And so you can think of this as a between and a within system neuroadaptation in a sense, but we know now, and we've known for some time, that withdrawal-induced increases in dinorphin occur similar to that of CRF, and we know that kappa antagonists can block withdrawal-induced anxiety-like responses, and we know that kappa antagonists can block dependence-induced alcohol increases in self-administration, and we know that kappa antagonists can block stress-induced reinstatement. Parallel effects to what we see with blockade of CRF. And now there's a great deal of interest in the field in the potential for kappa antagonists or kappa partial agonists in the treatment of not only alcohol use disorder, but opioid use disorder as well. So finally, to wrap up this section on the negative emotional states associated with withdrawal, or what I call the dark side of addiction and alcohol use disorder, I want to talk about the interaction of alcohol with pain. And so we do congressional briefings for the Congress at NIAAA, and we had one on women in drinking a few years ago. And Martha Woodruff, who's a former public radio journalist and late blooming novelist, gave a great speech. And I asked her whether I could get a quote from her, and this is the quote. "I grew up professionally focused and personally adrift. There's a long history of depression in my family, and my childhood home was dominated by my mother's mental illness that was not recognized, let alone treated. Sometime in my mid-30s, I started using alcohol to provide relief from the pain of childhood damage and organic depression. Bourbon was the only thing that would make the loop tape of despair in my head pipe down." So I want to remind you that alcohol is a pretty good pain reliever, but unfortunately only at doses that basically reach the level of a binge at 0.08 gram percent, which is the legal limit. But if alcohol produces pain relief, physical pain relief, it also causes pain during withdrawal. And on the slide here, you can see on the left-hand side that the electric pain threshold in individuals in severe alcohol withdrawal was dramatically decreased. In other words, they're showing increased pain. And a thermal pain measure in a separate group of individuals correlated with their Beck Depression Index score of depression. So the more depression, the less tolerance they had for pain. And I show you on the right-hand side that this pain response, this hyperalgesia, can actually be reversed in rodents by administration of a CRF antagonist. It's not a complete reversal, but a partial reversal. And this is a study done by Scott Edwards in 2012. And so one of the arguments is that there's not only physical pain during withdrawal from alcohol use disorder, but there's emotional pain. And I alluded to this earlier, but the argument is that these within system neuroadaptations and between system neuroadaptations all contribute to what we call apparent tolerance, the reversal of the A process, but this prolonged and more dramatic increase in the B process, or the dysphoric state associated with withdrawal from alcohol when used excessively. And so there's a good deal of study on this going on at NIH and in the external community at this point, but we know that there is a classic pain pathway, the spinal thalamic pain pathway that goes up to the periaqueductal gray from the dorsal root ganglia to the hypothalamus, maybe onto the amygdala there, but also to thalamus and somatosensory cortex, and then eventually back down to the amygdala. But there's another pathway that was described by Jean-Marie Besson in Paris and Donald Price at Hopkins that goes directly from the dorsal root ganglia to the parabrachial nucleus to the amygdala. And if you overlap the extended amygdala with the pain pathways, you see where the correspondence is actually in the central nucleus of the amygdala. And so the argument is that there's probably overlap between physical pain and emotional pain and physical pain, and that some individuals may ultimately be self-medicating alcohol to address this emotional pain that's associated with chronic administration and withdrawal. And that weaves this negative reinforcement into another framework of describing negative affect. I want to mention also that if one drills down into the molecular biology and epigenetics of alcohol use disorder in animal models, one sees also that there are molecular changes. And this is the work of Subhash Pandey and his group, where acute alcohol exposure can actually change the expression of genes by inhibiting histone deacetylases. And that's over on the left-hand side, and basically relaxing chromatin. But with chronic alcohol leading to withdrawal, chromatin becomes condensed and HDAC activity is increased, leading to this condensed chromatin and decreased expression of these genes, as well as decreases in dendritic spine density. And what Pandey has gone on to show with his group is that this alcohol exposure itself can lead to these epigenetic changes, and they're even more pronounced if the alcohol exposure is during adolescence. And so in some sense, by exposing young people and to alcohol early on, as we know, they are much more likely to develop an alcohol use disorder later in life. But the argument is that they would be affecting some of these same dark side circuits and setting them up for later dysregulation and neuroadaptive changes that contribute to the dark side. And so what I've said is that there are neurotransmitter systems that get recruited in the stress system to drive alcohol use disorder. They converge on the central nucleus and the amygdala. I've talked about CRF and dinorphin, but norepinephrine, vasopressin, hypocretin, substance P. I talked about glucocorticoids, but also a burgeoning area of interest is in neuroimmune factors that can interface with this neurocircuitry. And I haven't talked about the fact that there are buffers, stress buffers, homeostatic buffers, if you will, neurotransmitters such as NPY, nociceptin, endocannabinoids, yes, endocannabinoids, and oxytocin that may also become dysregulated. So if you can imagine stress transmitters being activated and anti-stress neurotransmitters being inhibited, you have a powerful dysregulation of the stress axis in the brain. So I'm going to finish by talking about the preoccupation anticipation stage. And this is an area where there's a good bit of work now in trying to understand how do these circuits in the old cortex and the prefrontal cortex control these basic circuits for motivation that we've been talking about. And so one of the arguments is that there is a prelimbic pathway from the prefrontal cortex to the nucleus accumbens, and an infralimbic pathway, and an infralimbic pathway to the amygdala. Now, a lot of people have been studying the prelimbic pathway and glutamate activation that leads to craving-like responses. And less study has gone on to the control by glutamate of the amygdala system. But we do know that dysregulation in both of these systems can contribute to protracted abstinence and craving. And I've listed 3 neurotransmitters here: glutamate, GABA, and ghrelin. Ghrelin is a hormone that's derived from the gut, but seems to be activated during craving for alcohol. And in human studies, there's been a good bit of work to show that frontal striatal connectivity and cognitive process is related to alcohol misuse, and particular areas that are relevant in the human condition is dorsolateral prefrontal cortex to caudate in goal-directed behavior, but also cue reactivity that involves the orbital frontal cortex projections to ventral striatum. And then, as we've already discussed, the habits with prefrontal cortex and also dorsal striatum being involved, that circuitry in humans. And then there are negative emotional circuits, as I discussed, involving possibly the ventral medial prefrontal cortex and, again, orbital frontal cortex controlling ventral striatum, but also, as I mentioned, amygdala. And we know that gray matter volume actually deficits predict the time to relapse in alcohol-dependent patients. And so in this study, 45 abstinent alcohol-dependent patients in treatment and 50 healthy comparison subjects were scanned once during high-resolution structural MRI, and this was used to assess regional brain volume differences between the groups. Significantly smaller gray matter volume was seen in the alcohol-dependent subjects in 3 regions: the medial prefrontal cortex, the right lateral prefrontal cortex, and posterior region surrounding the parietal occipital sulcus. Smaller medial, frontal, and parietal occipital gray matter volumes were predictive of shorter times to relapse to alcohol use in heavy drinking relapse. And you can see here in the proportion of patients without relapse, and the patients with smaller frontal cortex were more likely to relapse. This is a human study suggesting that disruption of function in the frontal cortex can lead to relapse. And I mentioned that glutamate is dysregulated. Over the years, there have been studies showing that glutamate is activated in animal models, but also in humans during ethanol withdrawal. And I urge you to focus on EW, ethanol withdrawal, in this study from Ronja Spanigel's group. But also, Marcus Heilig has shown that in a study done at NIH that a camprosate, which is a treatment for alcohol use disorder and known to reverse the glutamatergic surges that one associates with alcohol withdrawal, actually does so in detoxified individuals, human beings, treated with a camprosate. And you see here a dramatic decrease by day 25 of individuals being treated with a camprosate. So again, and we know that a camprosate is effective in reversing self-administration of alcohol in alcohol-dependent animals, the original study from Lemanian up in the left-hand corner, and also by Ronja Spanigel's group showing the alcohol deprivation effect is reversed by a camprosate, but in a whole series of human studies done in Europe, you can see increases in percent abstinence in individuals treated with a camprosate. Clinical studies show that a camprosate was effective in potentiating and facilitating complete abstinence from alcohol. So again, translational work suggesting that the original animal studies done in 1987 and then later studies done led to the approval of a camprosate as the second major treatment for alcohol use disorder. Again, I think lending to the argument that understanding the neurobiology and the neurochemistry and the neurocircuitry of alcohol use disorder has led to a number of treatments for alcohol use disorder and relieving of suffering for a great many people. So finally, I'm just going to wrap up by saying that I've argued over the years for not only homeostatic changes in neuroadaptation, but allostatic changes in neuroadaptation associated with alcohol use disorder and drug addiction in general. And so the idea is that the opponent process remains intact in individuals with alcohol use disorder, as illustrated by the opponent process here, but my argument is that there is a gradual shift in the allostatic set points such that instead of returning to homeostasis, hedonic set points drift lower and lower. And so you end up self-administering alcohol to fix the problem that alcohol caused. In other words, you're trying to claw your way back to a normal hedonic state. And so you've drifted away from homeostasis, but now you have an abnormal set point. And to make a comparison, think of obesity and think how easy it is to gain weight and how difficult it is to lose that weight once you've gained it. And then, as with any other chronic disorder, there are loads on the system, which we call allostatic loads. There could be genetic loads and epigenetic loads, as I described. There could be childhood trauma. There could be psychiatric comorbidities. But I note here that one of the major weights pulling down this system is excessive drinking. Finally, I'd just like to mention that some of the things we've been talking about have been impacted not only by the deaths of despair and interface with the deaths of despair, as I illustrated earlier, but also by the COVID-19 pandemic. And so we know that during the COVID-19 pandemic, there's been physical isolation and resulting social isolation. And it could be that some of the increases in drinking and some of the increases in drinking to cope during the pandemic also interface with the hyperketifia and the withdrawal negative affect states that I discussed earlier. So in conclusion, the neurocircuitry of alcohol use disorder can be heuristically studied within the framework of a three-stage cycle driven by multiple sources of reinforcement and motivation. The binge intoxication stage reflects a dysregulation of incentive salience and pathological habits mediated by neurocircuitry of the basal ganglia and includes activation of opioid peptides and dopamine neurochemical function. The withdrawal negative affect stage reflects the dysregulation of reward stress systems mediated by neurocircuitry of the extended amygdala and includes loss of dopamine neurochemical function and gain of brain stress neurochemical function, such as corticotropin releasing factor and dinorphin. The preoccupation anticipation stage reflects a dysregulation of incentive salience pathological habits mediated by neurocircuitry of the prefrontal cortex, and also includes activation and dysregulation of glutamatergic neurotransmitter function. Significant overlap in the engagement of brain circuits mediating negative emotional states, hyperketifia, and pain may be hypothesized to explain the role of alcohol and opiates in deaths of despair and possibly even contribute to the increases in drug seeking and drug taking observed in the COVID-19 pandemic. Thank you very much.